Hi, my name is Mike. I'm a research fellow at the Diamandis lab. Welcome to NeuroIQ. I'm just going to do another episode for this week. And I will be talking about what we're doing in the lab, specifically with the brain, as you know, and the diseases. Specifically, I will talk to you about some recent work we did with meningioma. So what are meningiomas? Meningiomas are a very common brain tumor type, one of the most common ones. They arise from the arachnoid cap cells of the leptomeninges. These are the layers that kind of cover the brain and protect it from the outside environment. Usually meningiomas primary tumors, and they are resected with surgery. Also sometimes they can be treated with radiation depending on the behavior that might be observed from doctors under the microscope. So what is the current issue with meningioma treatment? Nowadays doctors try to predict the behavior of meningioma under the microscope. They look at the specific traits to see and predict whether those meningiomas will recur. And currently this technique is not very efficient. So many times there are patients that are treated based on the technique and tool because there is a worry that the meningioma might recur, where actually this is not the case. And either patients do not get the treatment that they need, which is usually radiation, to kind of halt the recurring meningiomas. So currently there's a lot of interest and a lot of research groups that are looking into developing new tools to better predict this behavior of meningioma, specifically whether they will recur or not. So what kind of new technology we're using in the lab to address this need for new tools? We're using a technology called Mass-spectrometry. Mass-spectrometry is a very powerful tool that looks into protein patterns on a global scale. In other words, thousands of proteins are being profiled at any one time. We use this technology in a recent paper on meningiomas to try and find specific protein patterns and also the biology of those patterns to see whether the aggressive behavior of meningiomas could be seen or detected on the protein pattern level. And as a matter of fact, we published our results recently, and we did see a specific pattern developing with aggressive meningiomas, regardless of whether they recurred or whether they had a different WHO grading system that was specific to the aggressive behavior of meningiomas, namely their propensity to recur again and again. We're equally excited about another aspect of this research, which is basically the biology of these behaviors, of these tumors. So instead of just looking at the proteins themselves, we look at the whole biology of this behavior of meningiomas to try and figure out which pathways or which processes these tumors are addicted to, so we can target those pathways, we can inform therapeutics research, and hopefully try and devise a therapy that is much more effective to each meningioma patient. It's more kind of personalized to the behavior of each individual tumor, and hopefully it's more like an oral pill. It's nothing as invasive and destructive as surgery or radiation. So that was it. Hopefully you learned something about meningioma and how we are using new tools to find new treatments and better predictions. I would like to thank our sponsors, the Ontario Institute for Cancer Research, for providing funds for doing this project. So hit like and subscribe, and tell your friends about NeuroIQ if you want to learn more about the brain and the diseases and how it works.